Well, for me, it was kind of a long journey, to be honest. I started out in the public health sector, started out working in a hospital, a regulated hospital laboratory. And I guess that was where I met my first real mentor that really kind of persuaded me to go on in school, Dr. Kowalczyk, Dr. K as we called him. And he saw something in me, I think, that convinced him that he should write letters of recommendation for me to go on to graduate school. And so I think we've all had mentors over our career, whether it's in high school, college, postdoctoral, wherever it was, even department heads in various large companies. So I think I had a series of events working with various different people that led me into the medical microbiology field. I studied bubonic plague, of all things, Yersinia pestis, the causative agent of bubonic plague. And my professor at Michigan State saw my interest in medical microbiology, and he sent me on for an infectious disease postdoctoral fellowship. And from there I went into the vaccine field, preventive medicine, and I worked in vaccines and bioanalytical services for 25 years in large pharma. And recently, 6 months ago, I joined Frontage Laboratories to take my career experience from all of my large pharma experience and applying it at Frontage Laboratories to help the multitude of companies that Frontage services globally.
I think we made lots of contribution to the pharmaceutical society, and evidenced by we helped, and lots of pharmaceutical medicine product approved by US FDA and also another regulatory bodies. So I think that this is a major contribution from our team. Our team members feel very proud of it. Another contribution, I would say, Frontage Bioanalytical team become a major player in the bioanalytical space. And we are the one of the top bioanalytical lab in this area. I think our sponsor, our client, all appreciate Frontage's effort. And another contribution, I would say, is in the scientific contribution to the bioanalytical area, evidenced by many book chapters from our team, and also published the paper, and also attending all kinds of scientific meetings and post presentations. So I think that those are all we feel proud of it, and by doing this, at the end, help our patient, have the best medicine available and to the human health.
I think today is probably the most exciting time for new drug development. The technology that we're facing today to be able to measure things we couldn't measure historically. The sensitivity of assays that we perform today is 1,000 or even 1,000,000 times more sensitive than we've ever had. So we can identify new targets that we didn't even know existed before. And it makes it impossible to develop a drug if you don't even know what the target is. Now that we can measure these targets, we can actually identify drugs that are capable of blocking that target or interacting or enhancing that target. So having the technology to be able to measure ultra-sensitive, low-frequency markers not only allows us to develop new drugs to treat diseases, but also to monitor that disease, whether the disease comes back, the efficacy of the disease, could be a safety marker, kidney injury, liver injury, different types of things. So we can monitor the safety of the drugs to a higher level than we ever could before. We can measure the efficacy of the drugs to a higher level than we ever could before. And we can identify new targets that we never even knew existed before. So when we're getting into the immuno-oncology space, or even cell therapy or gene therapy, which are probably two of the hottest, fastest growing areas of medicine today, having this technology gives us the tools that we didn't have previously.
So this will be have more and more application in the pharmaceutical area to address emergent bioanalytical needs to address the challenge. For some of the large molecule quantification and also endogenous biomarker, they all need a high resolution mass spec to support. And that's one of the trend. Second, I would say intact molecule quantification using LC-MS, such as ADC, monoclonal antibody, fusion protein, oligonucleotide. All those has the unique challenge. And they have all kinds of approach to analyze those molecules in the biologic samples. But I think the intact molecule quantification, they have a unique role in addressing this kind of bioanalytical needs. Of course, they also have associated challenges with this new, new technology. For example, the sensitivity current high resolution mass spec is not as good as a triple quadruple mass spec. So I think the industry need to help how to improve those kind of challenges to meet the bioanalytical needs in the pharmaceutical industry. That's one area I think lots of lab in pharmaceutical company, in CRO business area, and they all pay attention and is working on this new technology. And another area I would say is microsampling. And in the past, dry blood spot technology got lots of attention. But recently, capillary tubing microsampling technology get more and more attention. I think that they have a unique advantage to help pharmaceutical industry to address some of the challenges associated with small molecule animal PK study and also pediatric clinical study. So with the limited sample volume, I think we do feel needs to have a microsampling technology available to support drug discovery development progress. And another final trend I would say is a paperless bioanalytical lab, or called electronic bioanalytical lab. As you know, bioanalytical lab, the key is the data. Every day we generate huge volume bioanalytical data. And so far, most of the lab, they have a hybrid system, meaning based on the paper documentation and the electronic storage system for the bioanalytical data storage, analysis, tracking, all those kind of process. In the future, I think electronic lab, paperless lab will be the trend. And this can really help us to organize your data, manage your data in the bioanalytical lab much better. Second is can significantly improve data integrity. So that's one of the big challenges in the bioanalytical lab.
So with regard to maintaining the most cutting edge technology for biologics, we know that there's been a rapid expansion of biologic drugs on the market in the past decade, and hundreds more are in development, and hundreds more will be hitting the market in the next few years. And trying to maintain the analytical capabilities to support all of these different types of new biologic drugs, not just monoclonal antibodies, but therapeutic proteins, or even cell and gene therapy, or immuno-oncology. These fields are developing very, very, very rapidly and showing a lot of promise. And having analytical equipment that's capable of providing the data to support these new types of drugs, and as John mentioned, the electronic notebook, and integrating that electronic notebook or that electronic work system with these new pieces of equipment requires a lot of interfacing. So it takes scientists, and it takes IT, and it takes engineers, and everyone coming together to integrate all these different technologies together to support. So no matter what the modality is, no matter what the drug modality, whether it's a small molecule or a large molecule, or even a gene replacement, where they're actually repairing a gene, for instance, like sickle cell disease, or some other gene where they can actually repair the gene in a person, requires us to be constantly pushing technology to support these drugs, because these drug advancements occur very, very, very rapidly. So Frontage Laboratories and other CROs are investing very heavily in technology to support this. And when it comes to the biomarkers can support biologic drugs or small molecule drugs. So again, having the technology that we talked about previously when I talked about ultra-sensitive detection, having the ability to measure a biomarker that might be specific for a disease, or it might be more generally applicable to a disease trait or a disease syndrome, having the ability to measure those and monitor those and provide that data to the client in an electronic form that can be submitted directly to FDA or EMA. Those are some of the challenges that we face, and having the right technology, the right scientists, the right IT people, and the right engineers all working in harmony together to meet the client's needs. Yeah, as we were discussing before about having IT experts and engineers and biologists and chemists all coming together in harmony to support, our programs, there's also a lot of regulation around the type of work we do. And attending meetings like the WRIB, which we're attending today, and other meetings that have a regulatory focus, not just a bioanalytical focus, but a regulatory focus, and interacting directly with regulators from US or Health Canada or World Health Organization, European agencies, all of those, and being aware. And being a global CRO like we are, we will do studies in almost every country in the world. And so we have to be familiar with the regulatory practices. So I think it comes down to a balance between science and compliance. And if your science and your compliance are matched together, then you've met the expectation going forward. It's a constantly evolving field, so attending meetings on a regular basis, interacting with our colleagues and our regulatory experts so that we know what they're expecting, and staying at the cutting edge of today's expectations and being prepared for tomorrow's expectations. I think that's what it's all about.
Frontage team use a client inspection, US FDA inspection, and other regulatory bodies inspection as a process improvement tool. For example, we have more than 12 US FDA inspection to our bioanalytical lab in US and in China. Every inspection we treat it very seriously, and even small suggestion, improvement action, we will go ahead to implement and follow up on this kind of thing. And all kinds of client inspection for all different purpose, facility qualification and the study-based inspection. And we use this as a continuous process improvement tool. The compliance quality is one of the most important area for the CRO business, and the Frontage pay huge attention on it.